What if I told you that researchers just figured out how to turn the proteins inside your living cells into quantum computers that could detect diseases before you even know you're sick? This isn't science fiction anymore. It's happening right now. And it's about to change everything we know about medicine and biology. But here's where it gets controversial. Are we crossing a line by turning our own biological systems into quantum surveillance networks? Here's the breakthrough that has the scientific community both excited and concerned. Maintaining quantum stability in ideal laboratory conditions is already incredibly challenging. Yet scientists have successfully engineered proteins to function as quantum bits, qubits, within living, chaotic cellular environments. The research team utilized fluorescent proteins as their quantum foundation because these remarkable molecules serve as natural nanoprobes that glow without requiring external dyes or markers. Through precisely timed laser pulses, these engineered proteins achieved quantum superposition states while simultaneously monitoring cellular abnormalities, potentially revolutionizing early disease detection methods. Imagine if medical diagnostics could evolve beyond their current limitations. Traditional disease tests can only reveal what's already happened. But what if we could create a real-time monitoring system that catches problems at the exact moment they begin developing? This futuristic scenario might become reality through proteins that operate using the same quantum principles found in cutting-edge quantum computers. Here's the thing most people don't realize. Biology and quantum computing are natural enemies. Quantum operations demand surgical precision and environments with virtually zero interference. Quantum bits, the sophisticated cousins of traditional computer bits, are notoriously fragile, even when housed in specially designed quantum machines with multiple layers of protection. This presents a stark contrast to the bustling, chaotic world inside living cells, where enzymes constantly trigger chemical reactions and cellular components race around performing countless biological tasks. But here's where it gets fascinating. Researchers at the University of Chicago discovered an ingenious workaround. They genetically modified fluorescent proteins to mirror naturally occurring proteins in human bodies. Then attach these glowing proteins to their target molecules and manipulated them to mimic quantum computer behavior. A molecule-scale genetically encodable qubit sensor could enable ultra-sensitive measurement techniques for fundamental research and medical diagnostics that are complementary to existing quantum sensing platforms. The research team explained in their groundbreaking study published in Nature Journal. Let me break down why this matters. Traditional computer bits exist in binary states, either on, representing 1, or off, representing 0. Quantum bits, however, possess an almost magical property called superposition, allowing them to exist in both states simultaneously. Despite containing just one bit of information, qubits can be both on and off at the same time. The electron spin within a qubit handles the processing work, but this only functions when the superposition of both states remains stable and the spin direction can change at lightning speeds. This is where fluorescent proteins become the heroes of our story. These remarkable molecules possess an almost supernatural ability to glow, thanks to fluorophores, specialized fluorescent compounds that absorb and emit light, which naturally form within specific amino acid structures. Scientists can attach these proteins to other biological molecules, creating powerful tools for observing cells at the molecular level. Unlike synthetic dyes, fluorescent proteins are preferred for studying biological processes like gene expression because they integrate seamlessly with living systems. The research team focused on enhanced yellow fluorescent protein, EYFP, which has been a laboratory staple for years. They discovered that EYFP possesses the perfect characteristics to function as a biological quantum bit. What makes EYFP so revolutionary is its stable triplet state, a quantum condition where two electrons maintain parallel spins while occupying different orbital paths. Within its fluorescent components, the researchers transformed EYFP into a functional qubit by bombarding it with ultra-rapid laser pulses that triggered superposition states. The electron pulse patterns reveal cellular processes with unprecedented detail and accuracy. When external factors disrupt the qubit's superposition, the disruption patterns translate into valuable information about the cellular environment, including abnormalities caused by genetic mutations. And this is the part most people miss. For the protein qubit to gather comprehensive information about cellular activities, scientists must genetically engineer the fluorescent protein to match the specific protein they want to monitor within a particular cell type. The glowing protein gets attached to its target protein and subjected to laser activation, achieving superposition and transforming into a nanoscale probe that detects cellular activities in real time. This process allows scientists to understand biological mechanisms, identify early signs of genetic diseases, and observe how cells respond to various treatments. But the implications extend far beyond medical applications. The researchers suggest this technology could revolutionize non-biological fields as well. Directed evolution on our EYFP qubit could be used to optimize its optical and spin properties, and even reveal unexpected insights into qubit physics. The research team noted, protein-based qubits are positioned to take advantage of techniques from both quantum information sciences and bioengineering, with potentially transformative possibilities in both fields. Here's where things get ethically complex. If we can monitor individual cells with quantum precision, are we creating a biological surveillance system that could be misused? Could this technology eventually lead to enhanced humans with built-in quantum sensors? And who controls access to this incredibly sensitive biological information? The scientific community is divided on these questions. Some argue that early disease detection could save millions of lives and revolutionize preventive medicine. Others worry about privacy implications and the potential for biological manipulation that crosses ethical boundaries. What do you think? Are we witnessing the birth of quantum-enhanced medicine that will benefit humanity? Or are we opening Pandora's box by merging quantum computing with our biological systems? Should there be limits on how deeply we can monitor and manipulate cellular processes? Share your thoughts. This technology will likely affect all of us, and the conversation about its ethical implications needs to happen now, before it becomes widespread.